Hello and welcome to AtheroTalk, the podcast series by the EAS Young Fellows, where we bring you closer to the people and ideas shaping cardiovascular science and atherosclerosis research. I'm Pelin Golforoush and I'm delighted to welcome you to this episode. Today, we're joined by Professor Manuel Mayr, British Heart Foundation Professor for Cardiovascular Proteomics and co-director of the BHF Centre of Research Excellence at Imperial College London. Professor Mayr is widely recognized for his pioneering work in integrating proteomics with other omics technologies, helping shift the field towards a more systems-level understanding of cardiovascular disease. Professor Mayr, it's a real pleasure to have you with us. Welcome to AtheroTalk.
Thank you. It's a pleasure to be here.
Let's start by getting to know the person behind the science. Could you introduce yourself to our listeners? Who is Manuel Mayr beyond the titles and achievements?
I was born in Austria, so I grew up in the Alps, and I've always really had a strong love for mountains and skiing. So while I was studying at university, I was actually working as a skiing instructor. And this has really stuck with me all my life. So every winter, we always go back to Austria for skiing. I'm now a father of 3 daughters. 2 have already left the house. Every time my daughter left the house, we bought a dog so that our flat is not getting too quiet. So I keep myself busy, but also grounded. And I have to say, I appreciate the dogs. They're always excited when you come home and they listen to what you say. This is not always what my daughters did, I have to say.
You trained in medicine, but relatively early on chose to focus entirely on research. Was that a clear decision from the start or something that evolved over time?
I would say it was a fundamental struggle. Obviously, if you do medicine, there's a very clear path ahead of you and you have a job lined up. So it didn't really seem prudent to leave Austria, leave medicine, and pursue research. But then on the other hand, you only live once, right? You should probably do what you're excited about in life. And so you can also take some risks. And I have to be really grateful to my wife because obviously we just had our first daughter. So we moved from Austria to the UK when she was 1 year old. So the relatives were not happy, especially the grandparents. But on the other hand, I think if there are opportunities, you need to grasp them and see whether you find your own career path. Because especially in research, there is no defined career path necessarily. You have to shape your own path. On the other hand, it is important to remain realistic about your prospects and be open to new opportunities also outside research.
That's a path many people wrestle with, so it's really great to hear your perspective on this. Looking back, were there any mentors or key moments that shaped the direction you took, especially towards proteomics and systems biology?
Yes, pretty much at every stage in my career. First of all, in medicine, we really had some professors, in particular Professor George Wick, who was at the time pioneering research into autoimmune responses in atherosclerosis. And he had really this ability to get people excited about science. And many scientists, like Joseph Penninger and so on, came from his lab. So I think it was this initial excitement that you get medics to be interested in research. And then my second key mentor was Professor Qingmuk Su. He's now back in China, but at the time he was also working with Professor Wick in Austria. And he then got the opportunity to move to the UK, asked me to come with him, and then persuaded me to take on the challenge of proteomics. The only problem was the only experience we had was Western blotting. So that was a bit of a hurdle to go from Western blotting to proteomics and systems biology.
Mentorship really does seem to play a defining role in scientific careers. For early career researchers listening, what advice would you give about finding direction in such a broad and rapidly evolving field?
Well, I think this is the excitement of science, right? It is never standing still. It's always rapidly evolving. And therefore, by definition, we cannot stand still. So it never gets boring. There's always a new challenge around the corner. But for me, the most important thing is to embrace new technologies. And obviously, there's a very famous quote from Sidney Brenner: progress in science depends on new techniques, new discoveries, and new ideas, and probably in that order. So we should not underestimate the importance of new technologies to gain new insights into disease and mechanistic understanding. And I think in the current times with proteomics, AI, this is especially true.
That's incredibly valuable insight, especially today. Your work has consistently moved beyond studying individual molecules toward integrating multiple layers of biological information. How has this shift from reductionist approaches to multi-omics and network biology changed the way we understand cardiovascular disease?
To give you maybe a context, when I was doing my degree, so this was pretty much my medical degree in the last century, uh, every professor was focused on one individual protein. And those professors were known for their work on this protein. They provided incredibly important mechanistic insights, but it was always in silos. And there are 20,000 proteins encoded in the human genome. So why should you dedicate your life to studying just a single protein? So I thought that these new technologies, and at the time they were very much in their infancy, opens us the opportunity to not just study what we can measure, but actually to go beyond, what we currently can measure, for example, with antibodies, and broaden our measurement capabilities by using mass spectrometry. Right, we have 20,000 proteins encoded in the human genome. We don't have antibodies for all these proteins. So therefore, people use the tools that are available, and certain proteins will be studied extensively, whereas other proteins will be simply ignored. So I think this is the excitement that we can move on from just studying individual proteins and ask the question, what is changing across the system? And in an unbiased way, ask the question, which proteins may be most important for the disease.
It almost feels like a shift not just in tools, but actually in mindset. With the explosion of omics data, one of the biggest challenges is interpretation. How do you approach translating complex data sets into meaningful biological or clinical insights?
Yes, this is a key point. I think generating omics data has now been democratized, and that's a good thing. But often it does help if you have an understanding of how this data are generated and have an expertise about the potential confounders in the data sets. So this is especially important if you do clinical proteomics, right? We've got many disease biomarkers that may be markers of sickness rather than the disease itself, or markers of medication that was given because of the disease. So I think it's important to realize the potential of proteomics, but we need to carefully evaluate the findings we get, and especially also have mechanistic follow-up experiments. So omics on everything is not the solution, right? You need to combine the omics data, use it as a foundation, and then work across many disciplines from bioinformatics with clinicians, molecular biologists. So you really translate these proteomic insights into mechanistic validation.
You've recently explored the effects of PCSK9 inhibition beyond LDL lowering, particularly in the context of acute myocardial infarction. One interesting finding was that many proteomic changes seem to be largely mediated by LDL reduction itself. Do you think this challenges the idea of significant pleiotropic effects of PCSK9 inhibitors, or does it instead refine how we should think about them?
Well, I think the findings for PCSK9 inhibitors were really incredibly clean, at least for apolipoprotein profiles. Most of the proteomic effects we see are indeed closely linked to the LDL particle lowering itself. That includes, of course, ApoB, but also many other apolipoproteins, such as ApoE and some C apolipoproteins that can also be found on LDL particles. And the lipidomic data go in the same direction. We're actually currently working on a follow-up manuscript where we use UK Biobank data to look at loss of function variants for PCSK9. And again, I think there is very good evidence that the key effects are indeed on lipid metabolism. So I would be very cautious about pleiotropic effects of PCSK9 inhibitors. Our proteomic data actually would argue that they are very targeted and very clean in their effects.
From your perspective, how close are we to seeing proteomics and multi-omics approaches influencing routine clinical decision making?
Well, uptake in clinical decision making is a long journey, right? We've seen that with LP delay, for example, right? I mean, lipoprotein delay is a well-known risk factor for atherosclerotic cardiovascular disease and aortic stenosis. According to Mendelian randomization, it's supposed to be a causal risk factor, right, for both diseases. And of course, we are awaiting the outcomes from the phase 3 clinical trials, right, whether this is really a new treatment target. But nonetheless, I mean, even as a biomarker, right, this has been proven over and over again that a one-time measurement of lipoprotein delay would be important to estimate your residual lipid risk. And still, it is not widely utilized. So I think we have to be realistic that proteomics will probably be more on the research side. However, the tools that are developed for proteomics, in particular the multiplex protein assays, I could see those assays actually translating into the clinic, right? Because right now you make a choice of which proteins you measure. I mean, every clinician gets a list of biomarker assays they can choose, and they need to make a selection, obviously, because of costs, because every measurement entails a cost. But if we now have the opportunity to measure multiple proteins at the same time, and measuring many proteins at the same time actually is cheaper than measuring individual proteins, then it may well be that we'll see a transformation how we do the biomarker assessment rather than using the single marker medicine from today to more kind of biomarker profiling with multiplex assays. And I think companies are working on this to replace the individual protein assays with multiplexed assays.
Could multi-omics approaches eventually redefine atherosclerosis as a heterogeneous group of vascular diseases rather than one unified condition?
Again, I think this is now exciting because, of course, we move beyond histology, right? We are not just looking at the lesions and classify them according to the histological stain. We're looking at molecular profiles. We are still hampered by the fact that we don't really have so much tissue available compared to, for example, researchers in cancers. But there are big initiatives going on with large biobanks where they do omics profiling at the RNA and the protein level, and heterogeneity starts to emerge. A key example are sex differences in atherosclerosis. We will know that men get atherosclerosis usually 10 years earlier than women. Yet women then catch up and the disease sometimes develops quicker. So we do see molecular differences at the protein level, for example, between female and male plaques that are linked to inflammation, but also to calcification. And I think these insights in heterogeneity in atherosclerosis can be very informative for future mechanistic work.
We are now able to generate extraordinarily complex multiomics datasets. But where do you think the real bottleneck lies today? Data generation, computational integration, or biological interpretation?
Um, it's a good question. Maybe on all fronts, but the biggest bottleneck is not the measurement, it's probably the meaning. And the future, I think, will depend on combining omics data sets from different molecular layers, right? Not just having an RNA-centric view, but actually having a validation at the protein level. Because at the moment, a lot of the biological interpretation relies on RNA measurements, and we know that RNA is not as close to function and the phenotype as the protein, and the correlation between RNA and protein on average is moderate to poor. So I think it will be important to add additional layers, use the RNA information in addition to the protein information. But again, if you work with clinical samples, I think the key is to have a strong clinical phenotyping and understand the potential confounders that could alter your measurements in, for example, plasma and tissue samples from patients.
Looking ahead, what do you see as the next major breakthroughs in cardiovascular research, particularly in the context of systems biology and integrative approaches?
I think a clear example is the proteomics in UK Biobank. I think you had a talk recently with Professor Sir Rory Collins, and he obviously has led the UK Biobank, and they've now managed to get funding to get proteomics data in 500,000 individuals. So this is unprecedented, right? We never had this amount of information available in cohort studies. And I think this is an exciting journey. There is a lot to learn during this journey, but it feels like the early days of GWAS. Right now, we are going into bigger and bigger studies, and our proteomic data become available at scale. That is basically unprecedented. And I think a lot of researchers will use this data, and a lot of discoveries will emerge from this data.
And as technologies evolve, how do you see the role of AI and computational biology shaping the future of proteomics and network-based research?
Well, I think will help. I mean, the challenge is that you cannot be an expert on all individual proteins. I mean, there are 20,000 of them. And, the key advantage, like we said at the beginning, was that people in their research career could focus on one protein, one pathway, and get really detailed knowledge. What are you going to do if you get proteomic profiles for 5, 10,000 proteins? How are you going to pursue all the candidates? And I think this is where really AI actually will be a great help, simply for literature mining, understanding, the connections between proteins, moving beyond what any individual researcher could probably interpret out of this proteomic data. So AI needs data. The proteomics and omics pipelines provide data. And again, in combination, I think we'll actually remove one of these bottlenecks, which is data interpretation.
AI-driven models may soon become extremely accurate at predicting atherosclerotic disease, but prediction is not the same as prevention. How can we make sure that AI helps us move beyond risk prediction toward true biological understanding and therapeutic innovation?
I think, again, you have got clear examples, right? I mean, if you look at AlphaFold, we never thought we could crack protein folding. AI actually did. Now we can make structure predictions. We can make new drug developments a lot faster using AI. And again, proteomic data can help there, right? There's possibilities for using cross-linking proteomics to study protein-protein interactions. And I think AI will just be transformative in how we can use this data to gain, more mechanistic understanding, which ultimately is key for therapeutic innovation. I think the risk is that we just do omics profiling at large scale data without the mechanistic follow-up experiments and the functional validation. So we should embrace AI, but we should also have a strong expertise in discovery science so that actually the findings that come out of the large cohorts can be taken forward into the wet lab experiments and the translational pipeline. So the risk, I think, at the moment is that data science is relatively easy because these data sets are out there, they're easy to use. But then the next bottleneck is how do we actually translate these findings into therapeutic innovation? And the risk is that we lose the wet lab science expertise.
It really feels like the field is moving toward a new level of complexity and opportunity. Before we close, we like to include a short reflective segment we call the graveyard of ideas. Science is rarely a straight line. Could you share a project or idea that didn't go as planned, but ultimately influenced your thinking or approach?
Um, I th- Again, maybe an example is the cardiovascular stem cell field. So in the early 2000s, everybody was doing stem cell research pretty much in cardiovascular research. Obviously, it was often not the real stem cell scientists that were doing this stem cell research. And if you go through the literature, it is obvious that some of these stem cells were not genuine progenitor cells. So one example from our own experience was that we worked on endothelial progenitor cells, which were cultured from peripheral blood mononuclear cells. And our aim was to use proteomics to identify surface markers that unambiguously identify those cells, because it was a culture method, but there was no individual stem cell marker to identify them. But instead of a stem cell marker, what we actually found was integrin alpha 2b. And integrin alpha 2b, as you probably know, is a very specific integrin that is only found on platelets. And that really got us thinking. And what we figured out is that obviously if people isolate PBMCs from blood, they're not just isolating PBMCs, they're also isolating co-isolating platelets. And if you seed those PBMCs in your culture dish, you actually have more platelets in the dish than you have PBMCs. So during culture, what happens is that the PBMCs, the monocytes, differentiate into macrophages. Macrophages like to eat up things. There's a lot of platelets around. The platelets die within 7 days, so they eat up the platelet debris. So if you then stain your macrophages after 5 to 7 days for endothelial markers, they stain positive because platelets share many markers with endothelial cells, such as CD31, van Willebrand factor. So you suddenly had hematopoietic stem cells staining positive for endothelial markers. They were then labeled endothelial progenitor cells. And nobody was bothered that they died after 7 days. That was seen as a culture artifact. And apparently nobody was worried too much that they didn't see mRNA expression for those endothelial markers. So again, it was something where we found, we looked for a stem cell marker, we found a platelet contamination. But it also taught me a lesson that you really need to challenge the conventional thinking in science. Because if you look at the literature of endothelial progenitor cells, right, you find thousands of publications. And, again, this is nice about proteomics. It doesn't just, show you the known unknowns, but the unknown unknowns. We didn't think about platelets contaminating these cultures and, EPCs masquerading as stem cells by incorporating platelet antigens into the macrophages. So again, this, I think, shows the power of proteomics. It was difficult to publish at the time, but obviously, I think, it stood the test of time, and it was ultimately published in Blood. Many high-profile journals who published APC papers didn't really want to publish the negative findings. So again, I think this was an important lesson. But I think, science moves on, and it's important to learn from our mistakes.
Thank you for sharing that. It's a powerful reminder of how progress really happens. One final question, particularly for younger listeners. If someone is interested in working with your group or collaborating, what's the best way to reach out?
Unfortunately, I have to say it's still by email. Having said that, we get quite a lot of emails these days. So please do email me again if you don't get the response. But otherwise, of course, in-person meetings at conferences would be great. There's, of course, the European Atherosclerosis Society meeting every year, but there are many other cardiovascular conferences. So if I'm there, please feel free out, to meet up.
That's very helpful. Thank you. Professor Mayr, thank you so much for joining us. This has been a fascinating conversation, both scientifically and intellectually.
Thank you very much. It was a pleasure.
And to our listeners, thank you for tuning in. If you enjoyed this episode, please subscribe to AtheroTalk, leave us a review, and share it with your colleagues. Until next time, stay curious, stay connected, and thank you for listening to AtheroTalk.